Assalamu alaikum. So what I'll do is go over just two slides. I realized and I was told that the second slide does not have any audio associated with it. So I'll start with the cloning, the idea of cloning. So basically what we do is that we take a gene from a human cell, and we don't have to take it from the DNA itself, from the genome itself or the chromosome itself. Rather, what we do is that we take the messenger RNA, we convert it to complementary DNA using an enzyme known as reverse transcriptase. And then the reason why we do this is that now the mRNA and its product, the cDNA, does not have the cDNA does not have introns, so it's the messenger RNA is already spliced. So we take this cDNA and we cut it with a restriction enzyme like ECOR1, for example. Okay. We take also a plasmid from bacteria, and we can purchase this plasmid from companies, by the way, and we open up the plasmid using the same restriction endonuclease, okay, like ECOR1. The reason why we use the same endonuclease is that the sticky ends of both the plasmid and the cDNA, now they can be complementary to each other. So we mix them and we close up the plasmid with the insert inside attached to it. First hydrogen bond, and then we use DNA ligase to close the gaps and form a phosphodiester bond. Then we take the plasmid with the insert inside, and we place it inside bacteria, and bacteria can clone it for us. Okay, now the idea now here is that we make copies, many copies of this insert this way. Now, we can take advantage of plasmids, since plasmids themselves, they contain genes, by the way, okay? So we can take advantage of plasmids and use them as expression vectors. Meaning what? Meaning that we place a gene and we put it in a plasmid, and we make bacteria synthesize or transcribe the gene and produce a protein from that gene. So we can make bacteria produce human proteins. And this is true for proteins like insulin, growth hormones, plasminogen activator, for blood clotting, and so on. So the idea is that we can produce large amounts of human protein using bacteria. We can take this protein and we can use it in medicine to treat patients like insulin for diabetic patients and so on. Okay, so this is just an example of bacteria that can produce a protein. So this is known as Western blot, which is similar to gel electrophoresis for proteins. So the idea is that we isolate all proteins from bacterial cells, and these are all of the proteins the proteins that we can detect from the bacterial cells that do not have this vector. And right next to it is proteins taken from bacterial cells that contain the plasmid and express the human protein, whatever protein is. And you can see that you can see the same bands, that is, you can see the same pattern, same proteins, okay, except for this one. And this is the protein that we force bacteria to synthesize. So you can see how much protein there is compared to all of the other proteins. So how can we do this? Well, we do it using plasmids known as expression vectors. Now, these plasmids are similar to the plasmids we use for cloning. So they do have an origin of replication. They do have an antibiotic resistance gene to select bacteria that would possess or contain the plasmid. And they do have a cloning site as well. Okay, so very similar. That is a site where we can use an endonuclease to open up the plasmid. Now, but this plasmid, or these vectors, these expression vectors, also contain three other components, and they are a promoter sequence. And this promoter would be upstream of where the gene would be inserted. And remember, upstream means in front of or before. Okay, so we use a promoter. It doesn't have to be a human promoter, by the way. It can be a bacterial promoter, but it has to be an efficient promoter. That is a promoter that is not blocked or inhibited, so that there is always production. There's always transcription and then translation. Okay, so that's one. We also need a transcription termination site right here. So when we insert the DNA gene, oh sorry, the DNA insert or the gene, you can have transcription starting at the transcription start site right here, and then you have a termination sequence. If you remember the stem loop structure, okay, so that you can terminate transcription. Once you have this messenger RNA, you need a third component, and that is the Shine-Dalgarno sequence. This is where ribosomes bind to at the 5' end, and then they search for the first AUG to start translation. Okay, so we do need a Shine-Dalgarno sequence. So these are the expression vectors. Okay, that's what they are, and this is what we use them for. Okay, so